Okay, so we're going to get started with this next session at PSB 2021. My name is Kristen Passrow, and I am a graduate student at Penn State, and I will be introducing to you this next session, What About the Environment? Leveraging Multi-Omic Datasets to characterize the environment's role in human health. So to start off, we're going to say something that everyone probably knows, but it's that the environment does play a role in disease risk. And one of the main questions is, how much of a role does it play? So back in 2016, a scientist attempted to estimate how much disease risk was due to genetic factors versus environmental factors. So they were looking at shared disease risk in monozygotic twins. Shared risk would mean genetic influences or shared environmental factors. But what they found when they estimated this was that it varied. So for different diseases, they may have a very small proportion of shared risk, which is a proxy for genetic risk, or they may have up to 50% of disease risk attributed to these shared factors. But what this means is that a substantial portion of disease risk is not due to genetics, it's not due to a shared environment, but it's due to some sort of unique environmental encounters that one person may experience and a person may not experience. And that may be contributing to different health outcomes. But when we talk about environmental contributions to disease, what is it that we are actually talking about? So to this end, I'm going to introduce the exposome concept. Now, the exposome concept is analogous to the genome. It's the totality of exposures that a person may encounter throughout their lifetime. So if we have an individual, we can group their exposome into three broad categories just for organizational purposes. But the first category would be internal exposures. These comprise the internal environment of the body and might have an effect on the cellular environment of a person. And these would be things like pro-inflammatory molecules, your microbiome, or reactive oxygen species that might interact with and affect the cellular environment. Now, another type of exposure, are specific external exposures. These are more classic exposures that you might see in a health study. They're things like diet, pollutants or toxins that someone might encounter, air pollution, and other material exposures you might encounter due to your occupation. Now, the final classification of exposure is something called general external exposures. These are very broad exposure. It's the broadest level of exposure. And they'd capture wide environmental factors, such as your geography, the climate, the weather, maybe your home environment. Do you live in a particularly rural versus an urban area? What is your socioeconomic status? Do you have access to education? Do you have access to healthcare? And also factors of the social environment. So interpersonal exposures, things like your social support, your relationship satisfaction, or maybe your work-life balance, things that would cause psychological stress on you that might in turn affect your health. So all of these three broad categories comprise the exposome. And another aspect of the exposome is that it is dynamic and cumulative. So it's constantly changing throughout your lifetime, and it starts in the prenatal period and it extends all the way until the individual's death. So it is a tall challenge to it's quite a challenge to try and sample this and measure this whole entire exposome of a person. But a study did attempt to take a snapshot of people's exposomes. Back in 2018, Zhang et al. Used wearable sensors to monitor people across time, in some cases up to 2 years. And they looked at the biological and chemical environments of these individuals, tracking them as they went day to day in their life, and they took samples of the microorganisms and the chemicals that they encountered. And what they found reflects back on this idea of the exposome as a very diverse, dynamic concept. For example, when they analyzed the microorganisms that these people were encountering on a daily basis, they found it varied widely from day to day. And there were many different species that they'd encounter, and it would depend on the location and the season as to which species were more abundant. They also found that each person's individual exposome was very unique, even if they were in generally the same geographic location. So all these factors, they were very personal to the individual depending on their lifestyle. And they found that the exposures, the abiotic and the biotic factors that they were sampling from these individuals' environment depended on things like the season, their geographic location, and in some cases, even the weather. For example, different chemicals were more prevalent before or after a rainstorm. So all this just goes to show the challenges that come from trying to measure and analyze something as broad as the exposome. In part because underneath this umbrella of the exposome, we also have different omics, such as the metabolome and the microbiome and the epigenome. So you could do an exhaustive study of any one of those omics and you'd still not have the whole entirety of the exposome. And again, that aspect of the exposome is something cumulative, something that's longitudinal and just constantly changing over time. That makes it difficult to analyze and to measure. And lastly, when it comes to actually measuring the exposome, typically you might think of health questionnaires or biological samples as sources of exposure data, but you could also find it in novel places such as expense records, maybe your social media or wearable sensors or home sensors. So all this is to say there is a lot of room for innovation and a lot of room for diverse applications of maybe developing open source software, developing novel approaches, untargeted approaches, agnostic approaches that all will improve environmental health research. And to this aim, we have we have organized this session for you to talk about how the exposome, how environmental factors influence human health. Now, a brief overview of our session schedule. We have two presenters today. The first, Christos Makridos, is going to discuss how the social environment affects COVID-19 outcomes and draw attention to how community-level environmental factors are pivotal in predicting health outcomes in different areas. And next, we have Morris Aguilar, who is going to explain a metabolomic pipeline he developed in order to analyze NMR data. And his research exemplifies how the internal and external exposures interact and influence one another. Now, please note that after each of these presentations, there will be a 5-minute live Q&A where you can post questions to the presenters using the Q&A icon at the bottom of your screen, and we will relay them to the presenters and they will be able to give live responses. After these talks, we will move over to a discussion section, and note that this will be in a different Zoom link, which we will post in the chat for you to link at the end of this seminar. We're doing something a little different. For our discussion section, we've invited a speaker who uses network science to analyze the exposome, particularly the foodome, and he's going to talk about the application of his research. Now, lastly, I'd like to thank my fellow session organizers for being instrumental in organizing this process. And I would like to also extend a thank you to our conference organizers for giving us an opportunity to present at PSB 2021. And all of you authors and attendees, thank you for participating. And without further ado, we will get started with the first presenter.
Hello, thank you so much for the opportunity to participate in the PSB conference. This is joint work together with Gil Alterovitz and Anish Mudid. We are studying the role that socio-demographic characteristics play in trying to understand. And so we're going to be looking at the role that the social environment plays in mediating the transmission of the virus using various techniques from the machine learning literature. Again, there is a large literature on the medical factors and comorbidities, but a much smaller one on the social factors. So let's get started. While there's been this emerging literature in the past 6-7 months on these comorbidities, namely age and in particular how different vulnerable groups have been affected, such as veterans in the Wrench et al. Paper, as well as the race in the Pan et al. Paper, researchers have still struggled to obtain bias-free and externally valid predictions on representative data sets. We are going to address that in two main ways. First, we're going to be using the population of veterans as our primary sample. So again, we're not going to be looking at non-veterans, but veterans are a pretty important group, especially when you're thinking about individuals that might be more exposed because of certain medical conditions after coming back from war, or individuals that are in their later years and therefore might be more at risk. In addition to that, we're going to be focusing on sociodemographic factors that have often been overlooked when trying to model the pandemic and the spread. We are specifically motivated by papers that emphasize the role of social capital. These papers, including some from my own work, focus on social capital as a glue that binds communities together. And when there is not that glue, when there is not that trust, it means that individuals are more likely to not take hygienic practices. They're more likely to go into restaurants and bars and not wash their hands and do all sorts of things. And so social capital and trust is going to be one of the characteristics that matters. And we're going to proxy for a lot of that by just using very standard, publicly available social demographic factors, including the industrial composition, namely that if an individual's in the retail sector, they're more likely to contract an illness than somebody that is in professional services and is maybe going to be working from home. Similarly, income is probably an important characteristic because individuals that are higher income might have better access to healthcare services, might have access to better preventative medicine or preventative behaviors. So all these characteristics might matter considerably when trying to understand the transmission of the virus and mortality outcomes. We're going to use three main methods for facilitating this prediction on a sample of 122 veteran healthcare systems. We're going to use a very standard ordinary least squares or OLS estimator. We're also going to use a lasso estimator or least absolute shrinkage and selection operator. And then we're going to use lasso with a ridge cross-validation. We're going to show that the lasso with ridge performs the best. And I think one of the reasons why this is so is because we don't have a particularly large sample. So you can't just throw all the variables in. You have to use some sort of variable selection model to decide what variables are more important. We're going to show that sociodemographic features matter even more than the standard hospital features, and that this ridge with lasso performs the best. So what we're going to do now is go into the characteristics that we're going to be using in this predictive model, and then we're going to show you the results in the 6 minutes that remain. From the American Community Survey, which is administered by the Census Bureau, we're going to see a bunch of characteristics about race, population density, the share of individuals that are male, the age distribution, i.e. the share of individuals between ages 0 and 18, between 19 and 24, so on, the share that are married, the share of education, so people with less than a high school degree, some college or more, the income distribution, the poverty rate. All these characteristics are going to be hugely important for understanding healthcare outcomes. And of course, we're going to have to see coronavirus cases and mortality. We're also going to see hospital level features, i.e. The number of services that a hospital provides, the average satisfaction, and some others as well. While we have almost 1,300 healthcare facilities, we only have coronavirus cases and deaths at a HCS level. Now, we could go into some of the administrative data from the VA, but what we wanted to illustrate in this simple article is using publicly available data, you still find that the sociodemographic characteristics matter greatly, and we just wanted to make it as transparent and as replicable as possible. In terms of results, we're just showing you how the ridge CV versus the lasso and ridge compare across various indicators or various metrics of performance. And so you can see in panel A that the R squared for the lasso and ridge is higher. It's around 0.62 as compared to 0.56 with ridge. We're also seeing in terms of when you're looking at mortality outcomes, you also see much better prediction coming from the lasso and ridge versus just the ridge. Similar results when you go down to other metrics like the root mean square deviation or the mean absolute error. Although it really doesn't matter which metric you're using, we just wanted to emphasize that this one seems to be performing the best. In addition, we're also looking at what features are most important, because that's one of the main objectives of our analysis, is to decompose how much social demographic factors matter. And what we're showing here is the absolute value of the features in terms of importance. And I just want to point out a couple that might have a lot of intuitive interpretation. So the share of individuals in professional services is intuitive because these are people in remote jobs. These are individuals that are not going out as often and are at much risk to expose others or to expose themselves. Similarly, the age that is between 55 and 64 or over the age of 65, those are of course going to be predictive given the correlation between age and the spread of the virus. We also see other industrial composition and the share of people in college or more, the share of individuals that are Black. All these characteristics are all social demographic features, and they're predictive of the log number of coronavirus cases among veterans. We're also seeing something very similar when we look at the log number of deaths, and we're trying to predict that. We see that professional services comes in even higher. That's again because these are more remotable jobs, and so people are going to be going into the office less at risk. You see something similar for some of the other factors, namely the individuals that are high school dropouts. These are individuals that probably have lower income. They're probably individuals with they're less likely to have access to healthcare. They might be eating less healthily, and so on. And so, bottom line is that these social demographic features are mattering quite considerably. Poverty rate, we could go through the list. However, what's key is that these are coming out as even more important than some of the hospital level features like satisfaction, like the number of services that a hospital provides, and so on. And we think that this is going to be important for policymakers or health practitioners to take into account. So in conclusion, while there has been this proliferation of models that are predicting coronavirus outcomes, whether infections or deaths, we're developing a model that focuses only on socio-demographic factors on the population of veterans in the United States, covering almost 1,300 medical facilities. We are showing that these social demographic characteristics matter even more than hospital features, suggesting that these clinicians and state and local policymakers have to take these geographic factors into account when they're designing public health policies and when they're designing treatments. So with that, thank you so much for your attention. We look forward to any comments and questions that people might have. You can reach any of us via email as well if you have further comments that you'd like to take offline. Thanks again.
Okay, thank you for that, Christos. And now we are going to go into a 5-minute Q&A section. We do have some questions in the chat, so I will relay them first. The first question is, were there any clinical features available, like how many people had comorbidities such as diabetes, heart disease, etc.?
Yeah, no, that's a great question. So, and you can hear me all right? Yep. Okay, perfect. So there's the data set that we were using for the results that we presented here was an aggregation. So what we also have experimented with is splitting up within each HCS, looking at the share of individuals with certain health conditions. But the short answer is that these comorbidities actually mattered quite a bit when you're working at the individual level data. When you aggregate up to the HCS, it actually looks at some of these socio-demographics were highly correlated and actually more predictive of some of the illnesses or some of these comorbidities. On the second one, or let's see, I'm just reading it right now. Did he feature an ablation study of the percent predicted? No, we did not do that. That's a good point. We should we should probably take note of that. Yeah, no, that's a good point. But I mean, yeah, so I guess to recap, though, essentially what we were trying to do is run a horse race between some of the hospital level characteristics together with all the usual medical characteristics, and then try to isolate the role of sociodemographics. But that's a nice suggestion, Megana. Thank you.
Okay, and now we have another question posted in the Q&A. The factors you identified were largely consonant with previously published results, such as age, race, profession. Were there any surprises or not previously noted significant factors, even if they had a small contribution?
Yeah, no, that's good. So one is just like, I'm going to totally admit that there are a lot of papers out there, and we probably didn't even like see, I don't know, 80% of them. There's so many pandemic papers. So if there are papers that you feel are making like literally the same point, we'd love to know about them because we don't want to be redundant. Our understanding is that there weren't any that have combined socio-demographic together with hospital level characteristics. And so to us, it was actually a little bit surprising that these socio-demographics kind of out-competed some of the like whether or not the satisfaction score within a hospital was sufficiently high, and also some of the other industrial composition we just haven't seen that much discussion of in the literature. Regarding the John, your point about social capital. So this is a great point, and the short answer is we have piggybacked off of some of the work that the Joint Economic Committee in Congress had done over the past year under Mike Lee's leadership, where they put together an index of social capital that wasn't using as sophisticated latent factor models. It was using more just like taking the average across different metrics. And so I think you're totally right that a latent factor model where you have this kind of like hidden Markov process and like multiple measurements is a better way to do it. I spent a lot of my doctoral years trying to figure out how to do some of this, and ended up struggling quite a bit. So if you or somebody else actually knows these methods really well, would love to talk for 20 minutes or so or get some recommendations. And then an anonymous question of how do we deal with correlated covariates in these models? So as an econometrician, I'm more of an economist than I am kind of like a health scientist or a clinician for that matter, is we're always thinking about omitted variables, whether or not that unobserved error is correlated with some of the hospital characteristics or sociodemographics. One way that we've tried to gauge the role of omitted variables bias is by removing some of the covariates and then seeing how much some of the coefficients change. This is what's called selection on observables. And so you can gauge the degree of bias by varying what you do include as observables. So that's number one, is that we were looking at some of those diagnostics. Number two is that we were more trying to build a little bit of a predictive model. And so we don't want to take the coefficients too seriously because we weren't trying to isolate a causal effect. We more wanted to get an understanding of which explains more of the variance in our outcome variable, and that's why we use these kind of lasso and ridge estimators. I'm not sure how we're doing on time, if there's another minute or whether I'm running up against the clock, so I'll let you take over, Kristen.
Okay, yeah, so we have maybe 2 more minutes of questions. If anyone would like to pose any more, we do have time, and if not, we could always move on to the next presentation. So.
The one thing I'll say, in case if somebody does have a question, feel free to jump in. But one of the things that we're building now is because we use the geographic aggregation. We did that purposely because we believe that public health interventions depend not only on individual level behavior, but also the communal behavior. And that's where we were trying to make the contribution here. But I think on an individual level, we are looking forward to training some of these models, especially using the longitudinal variation. Megana, I'm just taking a look at this additional question. Thank you. Did you see the sociodemographic factors coming up to be just as important as within your hospital analyses? So these sociodemographic, when we aggregate up to the HCS level, we were actually finding that these sociodemographic factors matter more than the hospital. Now, that comes with an extremely important caveat. The hospital level characteristics that we have are not great. They're based off of an older survey from about 2-3 years ago. Our rationalization for this was that we think that some of these hospital measures are very persistent. Hospitals are not changing massively from year to year, but nonetheless, part of the reason why we're getting that result might just be because we don't have as granular and as high frequency hospital level variation as we would like. So with that, I'm probably running into the 2-minute. So turn it over to you, Kristen, and thank you again so much for your attention.
Yeah, thank you very much, Christos. Okay, now we're going to move on to our next presentation by Morris Aguilar.
My name is Morris Aguilar, and I'm an MD PhD candidate in Dr. Molly Hall's lab at Penn State University. And today I'm going to talk about semi-automated NMR pipeline for environmental exposures and how this led to new insights on the metabolomics of cigarette smoke exposure. We used nuclear magnetic resonance data from a previous study, and we obtained this data from the metabolites repository. The original study only quantified 3 metabolites within the scope of lipid metabolism, but we have slightly different goals. Our goal is to apply a new semi-automated metabolomics pipeline, SAMP, to quantify more metabolites. And as we're taking a semi-automated approach, whereas the original authors use commercially available GUI software. And one thing that's worth noting about this data is that age, BMI, and cigarettes per day weren't included within the metabolites repository, only gender was. And we ran a confounding test with the logistic regression, and we found that gender was not a confounding factor. So SAMP addresses the arduous task of metabolite identification and quantification. As I alluded in the previous slide, commercial GUI software does not have advanced editing features, and it doesn't automate iterative steps, and it doesn't record user set parameters, nor is it open source. But SAMP addresses these pitfalls, and also it increases transparency of user set analysis parameters, and it unifies and harmonizes existing open source packages for spectral processing and multivariate analyses. So this figure right here is for illustrative purposes only. And within the cleanup portion of the analysis, there are 14 different steps that have different parameters that need to be tuned. And this process alone is ripe for automation. So SAMP harmonizes open source NMR software for metabolic environmental exposure studies. So within this pipeline, the gray boxes represent data and the white boxes represent a software package. So in the bigger green box, the NMR data cleanup section, the package PEPS NMR written in R takes care of the cleanup. And in the blue box, statistical analyses and modeling gets taken care of by scikit-learn for random forest modeling, and the statistical tests get handled by native R functions. In the metabolite identification, R-Dolphin handles the metabolite identification and quantification. And also this metabolite data gets piped to scikit-learn for random forest modeling as well. And lastly, in the pink box, metabolite statistics, that gets handled by metaboloanalyst written in R. And it does our t-test and pathway enrichment analysis. So after we clean up our data, the statistically significant bins determine which regions of the spectrum are peak aligned. So in other words, these results tell where the automated aligner needs to look at within the spectrum, and the other regions are ignored. And what this gets you is fewer statistical tests. And when we narrowed in on the significant spectral bins with the peak alignment, we found these metabolites. So 75 compounds were putatively identified. And 6 of those compounds were significantly perturbed. So in this figure, the blue triangles indicate increased metabolite in smoker, and the yellow triangle represents a decrease within a smoker. And the 6 metabolites that are significant are above the red line, which represents Bonferroni adjusted significance. So the big arrow is pointing towards a notable metabolite, indole 3-propionic acid. And the reason why it's notable is because it has been documented to be a neuroprotective antioxidant, and it's most likely to be affected in smokers with atherosclerosis. And another notable metabolite that was significant is indicated by the blue arrow again, and the metabolite is called indole sulfate, and it's a known cardiotoxin and uremic toxin. And in a previous study, it was shown that indole sulfate is lower in smokers' blood serum, but here we found that it was increased. So with SAMP, we can generate random forest models. So we trained the random forest with spectral bins and with metabolic data. And these are the AUC plots of those models. And for each model, it had a 10-fold cross-validation. And the green curve represents the mean AUC across those 10-fold. And you can see that is above the red dashed line, which means that the model is performing better than random chance. And when we train the random forest spectral bins, we do it as a method of exploratory data analysis to see if this data is worthwhile. And with the model I was trained with metabolites, we see that the performance improves a bit. And we think that it improves because when we filtered out non-informative spectra, the ones that were non-significant, it filtered out the non-significant spectral bins and most likely metabolites that are not as informative for the prediction task or classification task between smokers and never smoker. And one interesting thing about the random forest is that we can analyze its decision tree. So the metabolites within the decision tree weren't significant with our univariate t-tests, but there are some notable metabolites within this decision tree. So right now, the blue arrow is pointing towards 2,4-dichlorophenol. And the reason why it's interesting is because it is a known hazardous air pollutant, and that pollutant is absorbed by the tobacco plant. And another metabolite that was notable within this decision tree is 3-nitrotyrosine. And the reason why it's interesting is because it is associated with vascular endothelial dysfunction in smokers. And another metabolite that was also notable within the decision tree was xanthinic acid. And the reason why it was interesting is because there isn't any literature indicating any association with smokers. And another reason why it's interesting is because it's the top level decision node. So in other words, this metabolite is used within this model to classify between smoker and non-smoker, even though there is no literature indicating that this metabolite is perturbed within the smoking group. Also with SAMP, we ran a metabolic enrichment analysis. So all the metabolic pathways that are above the black dashed line are statistically significant after a Bonferroni adjustment. And notably, we replicated a major finding in the original study, such as the amino acyl biosynthesis pathway being statistically significantly enriched, and also the pathways of histidine, purine, and beta-alanine metabolism were significantly enriched. And these three processes were documented to be related to damage to the lungs. So the novelty of SAMP relies in its semi-automation to find metabolites and promoting study reproducibility. So anyone can go back and look at the code and see how the user is making decisions, which is unlike commercial software available today. And one of the major limitations of the study is the scalability of SAMP. The native packages do not support parallel processing. And the next step is to test SAMP on other NMR-based studies to classify disease status. Also, it's worth noting that SAMP is disease and environmental exposure agnostic, so it could be applied to any type of disease or exposure. With that, I would like to thank the Hall Lab for their support and feedback. And also thank you for listening. Take care of yourself, and if you can, someone else.
All right, thank you for that, Morris. And now we are going to open the floor to questions. And a reminder, if you want to post a question, you may put it in the Q&A chat. Okay, we have our first question popped up. It says the major environmental sources of xanthines, which include, for example, caffeine in humans, are coffee, tea, and chocolate. Is it possible that smokers being more likely to drink coffee account for your results?
To be quite honest, I haven't heard of that association, and I haven't looked into the literature on that. But also, one interesting thing about the xanthine is that it's incorporated within the metabolism of nucleotides, the subunits that create DNA, and it is involved within the process of degrading DNA. And yes, while it is used in metabolism of caffeine, within this study, I can't really say anything for sure that drinking coffee can be taken into account. And also, as I mentioned earlier on, given that this was a public repository that anyone can download, we were only limited to data from the participants about their gender and whether or not they smoked. So it might be interesting to see if there's some correlated habits between smokers, and depending on not they might be drinking more or less coffee. But whether or not there is a direct association, I cannot say for sure.
Okay, are there any other questions?
It's not that there's open air. I guess I could probably just continue talking. So one thing that kind of makes metabolomics interesting, and this is a relatively new field for this symposium, is that I'm assuming that everyone here is familiar that with the genomic technologies, people use Illumina and PacBio, and they're familiar with the data formats like FASTA and FASTQ. However, in metabolomics, it gets much more complicated. Because here I presented nuclear magnetic resonance data, and that had its own proprietary data format. And you can obviously translate into a more open source format, but also people use mass spectroscopy. And there's many different variations of mass spectroscopy, and that comes with its own file types as well. And you have to use different packages to extract and unpack that data so that it could be used in open source software, so that one can do their analysis on. And also, as I mentioned before, the packages that I used, the authors of those original packages, they weren't in direct communication with one another, because obviously their projects were a big undertaking in and of itself. But given that these authors weren't communicating with one another, one thing that was missing was that the packages weren't formatted for one another, thereby causing more barriers for piping this data from raw data to gaining biological insights.
Okay, so we do have a question. It says, how much input to your pipeline is required to produce results? And what portions of the workflow require the most user input?
Right, so how much data input? Technically, if you want something to be like at least statistically feasible, at minimum, like maybe 30, N of 30. However, the more samples, the better. But with this pipeline, as mentioned before, as you continue to increase the scale of samples, the bottleneck is the scalability. That's just the raw code. Your compute time will just increase by a lot. But your second question, what portions of the workflow require more user input? And that mostly lies in the cleanup part of the analysis. In the talk, I showed this one very complex, convoluted image that I preempted by saying this is just for illustrations purposes only. Like, there might be up to 14 different parameters that you all have to tune to clean up the data. And the reason why I have to adjust these parameters is because it cleans out the noise from the data. And given that the technology is nuclear magnetic resonance, most of the noise comes from chemical and even quantum sources, which is not what I'm going to get into. But there's a lot of chemical and physical noise you have to filter out with all these parameters, and that's where it gets pretty subjective. And the tough part about this when trying to replicate someone else's results is that you don't get access to the exact parameters that she or he set. And a lot of these GUI softwares, they literally just have like a little slider bar where you click and drag, and wherever you slide your slider bar to, that isn't actually recorded. So whenever someone else tries to reproduce a study with the similar GUI software, they wouldn't know to what extent the parameters were set to either high intensity or low intensity cleanup. So that gets lost in the mix, and that's something that SAMP is trying to address.
Okay, I think that wraps up the time we have for this Q&A. If you have any further questions, you can contact Morris directly. And for now, we're going to move into the discussion section. So I believe in the chat, we posted the link to our discussion where Dr. Barabasi will be presenting, and we'll all move over there and reconvene. Thank you all for attending.